Emily was at the City of Hope, and she's recently moved to UCSD, and she's going to talk today about both areas, cancer microRNAs, cancer host interactions, and exogenous RNA. Emily, I turn it over to you.
Thank you so much, David. I would like to thank Tanya and David for inviting me. So as mentioned, we recently set up our new lab at UCSD, and I also got a new email address. I'm pretty sure it's posted out there if you need to email me for any additional questions regarding my work. So without additional introduction, I would start my talk today. So I will cover the function of both endogenous and exogenous microRNAs we're studying in the lab. And my training background is in breast cancer biology with a focus on the tumor microenvironment. So based on my previous work on cytokines-mediated interactions between cancer cells and non-cancer cells, we actually started to work with cancer cell-secreted microRNAs a few years ago. So as shown in my first slide, showing the typical composition of a tumor microenvironment, using breast cancer as an example, so we have apparently cancer cells which by themselves are heterogeneous. We also have different types of non-cancer cells, including epithelial cells, vascular endothelial cells, fibroblasts, as well as other immune cells. So we know the interaction between cancer cells and the non-cancer cells is highly dynamic, and it also responds to external stimuli such as therapy. So for this talk, I'm going to focus on extracellular vesicles, and many of us call them exosomes because the preparations we typically use in the lab certainly include exosomes. And we will focus on the particular function of these extracellular microRNAs encapsulated in extracellular vesicles in the adaptation of the tumor microenvironment as long as the growth of cancer cells. So similar to the situation in the primary tumor microenvironment, through secreting these extracellular vesicles carrying microRNAs and other molecules, cancer cells can also prepare a pre-metastatic environment as shown in the right half of this slide. So we believe this occurs at a pre-metastatic stage before cancer cells metastasize to a secondary organ, for example, lung, liver, brain, and bone for the case of breast cancer. And this preparation of a pre-metastatic environment is important for cancer cells because multiple work have shown that dissemination of cancer cells occur at an early stage in tumor progression. However, this is not sufficient for those disseminated cancer cells to survive in the circulation and further colonize a distant organ. So in the well-known seed and soil hypothesis, a permeating soil at a secondary environment is necessary for these metastasized cancer cells to grow. So we actually also spend a lot of time studying how cancer-secreted microRNAs prepare a pre-metastatic niche in order for cancer cells to grow. So in 2012, so several years ago, we started a pilot project by deep sequencing the circulating microRNAs we purified from breast cancer patients. So this formed the basis for our current mechanistic study focusing on both endogenous cancer-secreted microRNAs and exogenous microRNA, including the plant miR-159 I'm going to talk about today. So in this initial work, we, I think we can skip skip this slide. So just to quickly remind you, the circulating microRNAs are found not only in extracellular vesicles but also in other forms. And I would like to clarify here that our lab mainly focused on the extracellular vesicle encapsulated forms of microRNAs, although other labs have also shown that those microRNAs that are independent of extracellular vesicle but associated with protein carriers such as Argonaute-2 and HDL can also be transferred from cells to cells. So, and again, in this study, I will only be focusing on extracellular vesicles, which include a lot of exosomes based on our characterization using electron microscopy. So, and back to our initial study profiling the circulating microRNA in breast cancer patients. All the patients involved in this study had stage 2 or stage 4 breast cancer, means that none of them had distant metastasis. So all the breast tumors are at relatively early stage at the time we collected the serum. And all patients went to the clinic and had their serum collected. At that time, didn't have any treatment, hadn't received any treatment, so all these are pre-treatment serum from early-stage breast cancer patients. Starting from around 500 microliter of patient serum, we performed RNA extraction by using triazole-LS reagent. So as you would know, here we didn't, in this particular experiment, we didn't purify exosomes or extracellular vesicles for the deep sequencing analysis in this very initial experiment. So what we did is to purify all circulating RNA and subject it to select the deep sequencing with a focus on the small RNAs between 17 and 52 nucleotide long. The sequencing results show that almost half of the small RNA sequences can be aligned to mature microRNAs, so confirming that mature microRNAs represent a major component of the extracellular RNA or small RNA found in human sera. So using deep sequencing, we were also able to compare patients with different types of breast cancer and those with different clinical outcome. And similarly, using the same RNA samples, we could do qPCR to confirm the levels of selected microRNAs. So this was the very first project we did about circulating microRNAs. And starting from there, we, as mentioned earlier, we identify those microRNAs in the blood that are associated with the status of estrogen receptor, as well as HER2 expression, as well as other features of the breast tumor. So this indicates, again, that circulating microRNAs could serve as potential biomarkers reflecting the nature of the tumors. And in the next experiment, so we asked the question whether these exosomes secreted by cancer cells can be transferred to niche cells and both in vitro and in vivo. So this experiment was performed like several years ago, and as we all know now, many labs were able to show the efficient uptake of exosomes or extracellular vesicles by multiple types of cells. However, this was the initial result from our lab using our own model, and in which we purified exosomes from the conditioned medium of cultured breast cancer cell lines using normal memory epithelial cells as a control, and we can further label these exosomes with dye I, which is a fluorescent dye that labels the lipid component of these vesicles. And then we can incubate these dye I labeled exosomes with different types of cultured cells to determine their uptake. So those cell types that can take up exosomes at a high efficiency are likely affected by the cargo in exosomes. So based on this, we confirm that exosomes secreted by cancer cells can be taken up by multiple types of non-cancer cells typically found in the tumor microenvironment. So this is our in vitro result. In vivo, we saw similar but not identical results. So when we label exosomes similarly with dye and this time inject into the mice through the tail vein, we do find a significant fraction of lung cells exhibiting the dye signal, confirming the uptake by a subset of lung cells. And we can also section tissues and use cell type marker to further confirm the type of cells in that tissue that can take up exosome. Using liver as an example, we separately label the hepatocytes and the Kupffer cells using different markers, and from this picture you could see that the dye signals are exclusively detected in Kupffer cells, which are the macrophages in the liver, but not in those hepatocytes, suggesting that in vivo, exosomes do have specific homing properties, and in this case, the exosomes we use in the lab, those secreted by breast cancer cells or memory epithelial cells, at least in liver, do not affect hepatocytes but only go to the macrophages. In other tissues like lung and brain, we saw uptake by the major cell types in these individual organs. For example, exosomes can be detected, or exosomal markers can be detected in lung fibroblasts labeled with this FSP1 marker, and in the brain, we can detect exosome uptake in both astrocytes and neurons, although in this picture, I'm only showing the astrocytes uptake. So these results actually confirm that we could use this in vitro and in vivo model to study the endogenous cancer-secreted microRNAs for their potential role in regulating non-cancer cells and thereby promoting cancer growth and metastasis. So continuing the topic on endogenous cancer-secreted microRNAs, we then asked whether these exosomes carrying cancer-secreted microRNAs could facilitate metastasis in vivo. What we did is to treat the mice by tail injection of exosomes of different origins from breast cancer cell line or from a non-cancerous memory epithelial cell line. After 6 times of injection, we challenged the mice by intracardiac injection of luciferase-labeled breast cancer cells, and we monitored the development of metastasis. So this is a frequently used model to study metastasis with a focus on the later steps during cancer metastasis after dissemination of cancer cells into the blood, because that's what the intracardiac injection of cancer cells mimic exactly. So the result indicate that comparing to mice pre-treated with PBS or exosomes from the normal control cells, those mice that have been pre-treated with cancer-secreted exosomes indeed had earlier development of distant metastasis in the lung and brain, suggesting that indeed cancer-secreted exosomes can prepare these distant niche in order to facilitate distant metastasis. And this was further confirmed by quantitative analysis showing more luciferase signals and thereby more cancer growth in the lung and brain if we pre-treated the mice with cancer-secreted exosomes. Starting from there, we actually published our first mechanistic papers in Cancer Cell in 2014, focusing on the function of a selected cancer-secreted microRNA called miR-105. For this study, we first focused on endothelial cells, which showed efficient uptake of these breast cancer-secreted exosomes as indicated by this dye internalization by cultured vascular endothelial cells. Normal cell-derived exosome and cancer-secreted exosomes show a very similar dynamic in the uptake by endothelial cells. However, that have different functions in endothelial cells. So this is a migration transwell migration assay showing that cancer-secreted exosomes have the unique ability to induce migration in endothelial cells. And we further purify the RNA from these exosomes, and we transfect the RNA, and we all know this is a RNA pool, including all different types of small RNAs as well as larger RNAs as well. So we transfected transiently with this RNA. We purified from exosomes into endothelial cells, which indicate that the exosomal RNA was able to confer the pro-migrative function we saw with cancer-secreted exosomes. So we further focus on this miR-105, which is highly secreted by metastatic breast cancer cells. As indicated in this figure, when we compared a panel of breast cancer cell lines and the non-cancer cells, MCF10A, those breast cancer cells originally isolated from pleural effusion, indicating high metastatic potential, express much higher level of miR-105. And in the exosomes we prepared from these cell lines, those metastatic breast cancer cells also secrete a much higher level of miR-105 into the extracellular vesicles. So this indicates that miR-105 could be related to the metastatic potential of breast cancer cells. So from here, we further studied the recipient endothelial cells and confirmed that exosomes secreted by breast cancer cells can indeed transfer this microRNA miR-105 to the recipient endothelial cells, whereas the endothelial cells typically have a low endogenous expression of these microRNA. And the primary microRNA and the precursor of miR-105 are not significantly altered in exosome-treated recipient cells, confirming this transfer of miR-105 is indeed through exosomes, but not through any indirect mechanism that could regulate the expression level of miR-105 in endothelial cells. The reason we focused on these microRNA also include that this microRNA is predicted to target a tight junction protein called Z01. This protein is relatively famous in cancer study because it's a very important component of tight junctions, and also tight junctions have been shown to be tightly related to cancer cell migration and metastasis. So we found two conserved binding sites of miR-105 in the 3' UTR of tight junction protein 1, or TJP1, also known as Z01, and using the reporter assay, we confirmed that these two sites indeed mediate downregulation of the mRNA targets by miR-105. So the tight junctions play important role in both epithelial and endothelial cells as indicated in this picture. They're both barriers and fences in polarized endothelial and epithelial monolayers. So important functions of tight junction include limiting those proteins that are expressed on the apical surface from migrating freely into other compartments of the plasma membrane. And as a fence, the tight junctions also limit those proteins and molecules from one side of the monolayer from penetrating the monolayer and migrating to the other side. So as you could tell, it's very important because with the leaky monolayers, one would expect more cancer cell migration through the blood vessels and epithelial layers, which would promote distant metastasis. Indeed, we see that when we overexpress miR-105 in memory epithelial cells, we see this destroyed polarized structure. So this is cultured in 3 dimensions using matrigel as a matrix. As you can see, whereas the normal epithelial cells typically give this sphere, a mammosphere structure on matrigel. A highly invasive structure was observed with cells overexpressing this microRNA. And we confirmed the downregulation of the target gene Z01 at both the RNA and the protein level, and confirmed that exosomes can transfer sufficient miR-105 to recipient epithelial cells to significantly downregulate Z01. So similarly, in endothelial monolayers, we saw the significant downregulation of Z01 by exosomes carrying high levels of miR-105. And as indicated by this immunofluorescence image, you can see the downregulation of Z01 at the protein level and the internalization of another tight junction proteins from the junction areas to the intracellular compartments. So this is strong indication for destroyed tight junctions in endothelial cells. As expected, we also observed the increased leakiness of endothelial monolayers in vitro using this model. So we see that endothelial monolayers pre-treated with exosomes high in miR-105 have increased the leakage of the rhodamine dextran dye we added to the top chamber of this transwell insert. And this effect caused by exosomes with high miR-105, such as the MD231 derived exosome, resembles the effect caused by VEGF, a famous cytokine that is known to regulate the migration of endothelial cells. So further, when we culture endothelial cells in 3 dimensions in matrigel, comparing to normal endothelial cells treated with normal cell-derived exosomes, which still form these branchy 3-dimensional structures, the structure can be destroyed significantly by exosomes high in miR-105, suggesting the microRNA also act on existing vasculatures to downregulate the integrity of the vasculature. So in vivo, we also confirmed the leakiness inducing function of secreted miR-105. In this case, we also pre-treated the mice with exosomes high in miR-105, followed by injection of the same rhodamine dextran dye through the tail vein. And we then perfused the animal to remove all extra dyes in the blood and only focus on those that have penetrated the blood vessels in a given organ and indicating vascular leakiness in such organs. So as seen here, mice pre-treated with exosomes with high miR-105 indeed show significant leakage of the red dye into multiple organs, whereas normal mice or control mice treated with normal cell-derived exosomes still show a pretty intact vasculature in these organs. As to cancer metastasis and how it is regulated by miR-105, we use a poorly metastatic breast cancer model called MCFDCIS, and we overexpress the microRNA in this cell line. In mice, when we injected the cells into the memory fat pad, which is right here, this is number 4 memory fat pad, comparing to the control cells, which are, again, poorly metastatic, we see significantly enhanced metastasis to multiple organs when we overexpress miR-105 in the primary tumor. And we also see increased local invasiveness in the primary tumor expressing high miR-105, and we also saw increased vascular leakiness in those mice bearing high miR-105 expressing primary tumor. So this suggests that primary tumor can secrete sufficient amount of miR-105 to adapt the vasculature at a distant site. And this is consistent with the downregulation of Z01, the target gene of miR-105 in the endothelial cells found in these organs. In contrast, if we block the function of miR-105 in a highly metastatic breast cancer model, the MD-MB231 model, we actually see suppressed metastasis at week 6 after we inject the cancer cells into the mammary fat pad, comparing to the 2 control groups. Although in these mice that have received the anti-miR-105 oligos, we still see lymph node metastasis, as indicated in all 4 mice showing positive signals in the chest area in this experiment. So consistently, we saw downregulation of CO1 in the endothelial cells from cells bearing high miR-105 tumors, but when we treated the mice with anti-miR-105, we can partially restore the expression of CO1 as well as the integrity of the vasculature, as indicated by less leakage of the dye into these organs. Lastly, for this project, we compared the levels of miR-105 in the serum of breast cancer patients. So we used the patients I mentioned earlier in our initial study. So we had 42 patients with stage 2 or stage 3 disease, and we had the follow-up data with these patients. Some of them developed distant metastasis, whereas others hadn't. So with that information, we were able to compare the levels of circulating miR-105 in those patients who later developed metastasis versus those who without disease progression during the comparable follow-up time. The results show that circulating miR-105 levels are indeed significantly associated with the risk of metastasis in early-stage breast cancer patients. Further, when we compare the serum level of miR-105 to the tumor level of miR-105 determined by in situ hybridization, and further to the ZO1 expression level in the tumor determined by immunohistochemistry, we saw a positive correlation between the levels of miR-105 in the blood and in tumor, indicating the more the tumor cells express this microRNA, the more they can secrete into the blood. And in contrast, we saw a negative correlation between the levels of miR-105 and ZO1 expressed in tumor cells, confirming ZO1 is the target downregulated by miR-105. Further, when we look at the ZO1 expression in vascular endothelial cells adjacent to tumor cells, we also saw a significant negative correlation between the level of ZO1 and circulating or secreted miR-105, indicating the extracellular function of cancer-secreted miR-105 in regulating endothelial ZO1 expression. So we also used the 3D culture of endothelial cells, confirming that patient serum with high miR-105 level can indeed regulate the 3D branchy structure of endothelial cells. So we used the case of patient serum that is high in miR-105, and comparing to the control normal serum, we do see the regression of these branches from these endothelial clusters. And if we pre-transfected the endothelial cells with anti-miR-105 so that they're resistant to miR-105 induced effect, we do see resistance of endothelial cells to patient serum. And these are representative pictures showing the correlation between tumor-expressed miR-105, indicated by blue color here, and the expression of ZO1, indicated by brown colors in the tumor cells, as well as in the tumor-adjacent vascular endothelial cells. And ZO1, as reported previously by others, again, seems to be associated with disease progression in breast cancer. So that is an example I wanted to present today to show how the endogenous microRNAs, like including those secreted by breast cancer cells, play a role in cancer progression and metastasis by regulating neighboring or distant non-cancer cells. So in this model, summarizing the miR-105's function, we have recently reported, as we know, in normal mammary tissue, and again using breast cancer as a model. So we have both epithelial monolayers and vascular endothelial monolayers in the normal tissue architecture, and these monolayers form as natural barriers against metastasis. Because for cancer cells, for cancer cells arising from the luminal area to migrate all the way to the blood vessel and get disseminated throughout the body, they first need to cross both epithelial and endothelial barriers to arrive to the blood. However, in cancer, especially in those breast cancers highly expressing and secreting miR-105 into extracellular vesicles, these vesicle carrying miR-105 can first arrive to epithelial and endothelial monolayers, destroying the tight junctions in these barriers, thereby facilitating cancer cell migration and dissemination. In a metastatic environment, similar effects would be observed. So cancer-secreted miR-105 could be detected at a pre-metastatic stage, as indicated in our patient data. And this miR-105 can then arrive to and adapt a distant tissue, for example, lung or brain, downregulating the tight junction of endothelial cells in those organs, thereby making holes on the blood vessels and facilitating disseminated cancer cells to invade and colonize these organs. So with this, I would like to move on to the second part of my project, focus on the exogenous microRNA we have studied. So I have to tell you that we're not currently following this direction because my training background and the setup of my lab are really optimized for the study of cancer-secreted mammalian microRNAs. However, we recently published this plant microRNA paper in Cancer Research. I do think it's very interesting. And I would definitely be more than happy to stay in this area, at least to learn what other people have found about the cross-kingdom regulation and how these plant microRNA or other exogenous microRNA could affect human diseases, including cancer. So back to this study, the reason we start this plant microRNA project is, again, the deep sequencing results of circulating RNA we isolated from breast cancer patients. So following the initial report of dietary intake of miR-168 from rice, so I think we all know this paper from Chen-Yu Zhang's lab, we asked whether we could also see plant microRNAs or any foreign microRNAs in our deep sequencing database. So what we did is to went back to our patient serum deep sequencing data, remove all the sequences that can be aligned to human genome. So by doing that, I think we might be removing some microRNAs that are conserved between mammals and plants, for example. So we're really, only looking at what has been left after alignment to human genome. So using the using the small RNA sequences that couldn't align, we then try to align these sequences to the plant microRNA database, and this is how we focus on miR-159 in this project. So, and as you know, we have seen multiple reports in this area, and as this is such a controversial field, and we have seen findings support the dietary uptake of microRNA, and findings that really questioning this discovery. So I've listed a few papers that have made us think a lot here, but I'm pretty sure you would have your own favorite papers to be added to this list. So basically, after knowing all these arguments in the field, we still decided to follow miR-159 because we did identify the sequences, and as indicated in our paper, which I probably don't have in my slides, we saw a perfect alignment to plant miR-159 in using sequence identified from breast cancer patients, although the counts are really low, and we were also able to confirm by PCR that we can detect, and again, although at very low level, we can detect miR-159 in these patient RNA samples. So we thought, is there, let's see if it really does anything. So with that in mind, so we started to learn a little about plant microRNA, and as you all know, they're pretty different from mammalian microRNAs, and they have this 2' O methylation at the 3' end to increase the stability. They have different way to form complementary with endogenous targets, and normally in plants, the target sites for microRNAs are also in different locations within the gene. So in plant, most of them are in the coding region and the 5' UTR, whereas in mammals, most time we look at the 3' UTR. And we also did a little research on the function of miR-159 in plants, and we found this is a highly conserved microRNA in plants, and the function in plants includes regulation of growth and leaf morphology, and as you can see here, plants with miR-159 knockout have this abnormal leaf morphology, and they're also shorter than the wild type. So targets of miR-159 in plants have been identified, and those include MYC33 and MYC65. Both are important in lineage differentiation and growth. And for these two identified plant targets of miR-159, we couldn't find any homologs in mammals, and thereby we used our own way trying to find the potential mammalian targets of this plant microRNA. So what we did first is to confirm, as I mentioned earlier, in our breast cancer patient serum samples that these plant microRNAs can be detected. So in addition to the sequencing result, we performed Techman PCR, so which shows that comparing to serum from healthy donors, breast cancer patients overall have a lower levels of miR-159. And if we further look at those patients with or without disease progression during 5-year follow-up, those patients who had a worse outcome with the disease progression had even lower levels of miR-159 in the blood. In this experiment, we used mammalian miR-16 as the internal reference. I know that miR-16 has been reported to be related to certain disease. However, in our particular patient cohort and circulating microRNA data set, miR-16 is amongst the most consistent circulating microRNAs, and thereby we used this as the internal reference for several studies. Although currently we are adding more internal references to our ongoing projects. So further, we look at the compartmentalization of plant miR-159 using miR-16 as a control. We purified extracellular vesicles from patient serum by ultracentrifugation, and we also saved the supernatant as the non-exosome fraction and we measured the levels of microRNA in each fractions. So this figure shows the levels of extracellular vesicle-associated microRNAs in comparison to the total and unfractionated serum. So as you can see here, plant miR-159 is mainly detected in extracellular vesicle. We were able to recover 70 to 80% of overall circulating miR-159 from the purified extracellular vesicles. So this is pretty similar to the case of miR-16, the mammalian control we used here, for which we see around 80% localization in extracellular vesicles. So this again confirms the hypothesis brought up by others that plant microRNAs can be taken up by intestinal epithelial cells, which will then secrete extracellular vesicles into the circulation. Although we didn't do further characterization of these extracellular vesicles to see their to identify their producing cells, we do know that these plant microRNAs are somehow secreted, processed and secreted by mammalian cells, and this is how they are able to be further delivered to other types of recipient cells in mammals. So these are in situ hybridization, confirming that we can detect miR-159 in some breast tumor tissues, although in many other tissues the staining is super low. So here we should look at the blue color indicating miR-159 levels. And interestingly, although most tissues give pretty low level of miR-159, we do see a positive correlation between the circulating level of miR-159 and the intratumoral levels of miR-159. And the next thing we did is to confirm that human serum contains sufficient amount of miR-159 to regulate cell functions. I think this is critical because we were very bothered by the fact that the levels of miR-159 we detected is so low in the serum. Although we totally understand that circulating exosomes are highly dynamic. They are taken up by recipient cells from time to time, so their half-life won't be too long in the blood. We still want to have the confirmation and also to be able to focus on a phenotype for the miR-159 study here. What we did for this experiment is to first isolate the exosomes from pooled normal human serum, because we know that healthy donors have the highest level of miR-159 in the serum. So in this case, we used the pooled sera from healthy donors to isolate exosomes or extracellular vesicles that we think would be high in miR-159. So we extract RNA from these exosomes, and then we first confirmed that these the miR-159 we identified from the circulating exosomes are indeed from plants, but not any some contamination or non-specific detection from some mammalian genes. So we treated the RNA isolated with sodium periodate to oxidize the 3' end of this RNA. If the microRNA is from plants, it should contain the 2'O methylation at the 3' end, making them resistant to oxidation in this reaction. So whether the mRNA, the microRNA can be oxidized, can be further detected by can be further indicated by the subsequent PCR reaction, because the MI script, the PCR reverse transcription and PCR reaction would not detect the microRNA following oxidation of the 3' end. So using this system, we were able to compare the exosome fraction and the non-exosome fraction of the microRNA we isolated from human sera. So in this case, when we compare the plant miR-159, we see that the EV-associated miR-159 are totally resistant to oxidation, suggesting that they have the 2'O methylation and it's from a plant origin. In contrast, in non-exosomal fraction, a significant fraction of miR-159 is oxidized and cannot be detected comparing to the unoxidized control. So the case is quite similar for the other plant microRNA, miR-168, the first one that has been detected for the cross-kingdom function. Although miR-168 seems to have a significant fraction of degradation even inside of the extracellular vesicles, so it could be that the 2'O methylation for miR-168 is not as efficient as other plant microRNA, so that we have both methylated and unmethylated plant microRNA encapsulated in the extracellular vesicles. So the control is the mammalian miR-16, which we know do not have any 2'O methylation, and indeed this microRNA is super sensitive to the oxidization experiment. And we couldn't detect any miR-16 anymore after the treatment, so this serves as a good control, indicating the difference between mammalian microRNA and plant-derived microRNA we detected in human sera. So in the next experiment, we studied the function of miR-159. So in many of these experiments, we started by using a synthetic miR-159 with the same 2'O methylation to maximally mimic the plant-derived microRNA, and we transfected different types of cells with these synthetic microRNA using scrambled sequence as the control. So by doing this, we were able to show that miR-159 can suppress cell proliferation in the 2 breast cancer cell lines we have tested, MD-MB231 and the MCF-DCIS cells. When we look at normal cells, including MCF10A memory epithelial cells and the primary memory epithelial cells, we did not see such function with miR-159, suggesting this is probably a cancer-specific function that miR-159 can conduct. Sorry, this is a slide I was trying to introduce. So the next thing we did is to confirm human sera contains sufficient levels of miR-159 to inhibit cell proliferation. We isolated exosomes from human serum like mentioned before, and this time we treated the breast cancer cells MDA231 with either the locked nucleic acid version of anti-miR-159 to block any potential functions mediated by miR-159 in these serum exosome, or we transfected the cells with a control LNA sequence. And then we, following serum exosome treatment, we measured the proliferation of the breast cancer cells, and here is the result. So if we look at the control recipient cells, these are control recipient breast cancer cells, following human exosome treatment, we indeed observe a significant decrease in cell numbers, indicating an anti-proliferation function of exosomes purified from normal human serum. And of course, these exosomes include all different types of things, from mammalian protein microRNAs to plant microRNAs and to other proteins and RNA as well. And then when we overexpress this anti-miR-159 to specifically block any miR-159 mediated function in breast cancer cells, this time we partially block the anti-proliferative effect of purified human serum exosomes, indicating the miR-159 in human serum exosomes indeed contribute to the anti-proliferative effect of human exosomes on breast cancers. Next, we try to identify the mammalian targets of plant miR-159, and again, because the plant targets of miR-159 do not have any homologue in mammals, we had to start, the target identification process using our own methods. We actually used 3 different methods to find the real targets of this microRNA. The first one is the Diana micro-T version 3. So this is an online software that predicts the potential mammalian targets for any sequence, any microRNAs from any species. So the second method we used is a RNA trap functional assay in which we transfected cells with biotinylated synthetic miR-159. We also co-transfected the cells with a dominant negative form of GW182. This mutant would stabilize the complex of microRNA and its nature target mRNA, allowing us to identify the real targets inside of the cells that are at that time interacting with the biotinylated miR-159. So using this method, we pulled down the biotinylated miR-159 along with its trapped target mRNAs, and we did RNA-seq. And this allows us to identify, again, like almost 1,000 target genes that at that time are associated with miR-159 in the cells. The third method we use is the RNA sequencing experiment looking at the gene expression level in cells following transfection of synthetic miR-159. So this is another intracellular functional analysis at the gene expression level. So we focus on the targets that are predicted by all 3 methods, and these include TCF7, NCOA6, and EN2, these 3 genes. So we further confirmed that all 3 genes can, sorry, let me see. So we further confirmed that all 3 genes, 2 of the 3 genes showed downregulation by synthetic miR-159 when we repeat the synthetic microRNA transfection in different cell lines. And among the 3, we further focused on TCF7, which gave us the greatest fold reduction in all cell lines we have tested. And when we confirmed the risk trap results by doing reverse transcription and PCR detection for miR-159 binding mRNAs. We also confirmed that TCF7 is significantly associated with transfected miR-159. I mean, the TCF7 mRNA is associated with miR-159, whereas a control mRNA is not detected in the precipitants. So from there, we hypothesized that plant miR-159 regulates TCF7 and thereby regulating the downstream pathways regulated by TCF7 transcription factors. So here we're showing the complementation of plant miR-159 with the 2 putative binding sites in the 3' UTR of TCF7. Reporter assays confirm that both sites actually contribute to the downregulation of the gene by miR-159. And further, at the protein level, we confirmed the downregulation of TCF7 by transfected miR-159. We know MYC is a downstream target regulated by TCF7, and similarly, we saw downregulation of MYC in the 2 cancer cell lines we have tested, although we didn't see significant downregulation of either TCF7 or MYC in the normal cells, possibly because of the low endogenous levels of TCF7 and MYC expressed in these cells. This also partially explains why there's a cancer-specific anti-proliferative function of miR-159, and that is because normal cells don't express a high endogenous level of its target TCF7. We also used the TCF reporter assay, the famous top flash assay, to confirm the miR-159 also downregulate the transcriptional activity of TCF7. And EBOX is the reporter for MYC, and similarly, we saw the plant microRNA downregulate the MYC transcriptional activator activity. And the negative control used for this study is a reporter for for NF-kappa B, and we didn't see any effect of the microRNA on NF-kappa B mediated gene transcription. In the next experiment, we restored the expression of TCF7 using a version of the gene without the 3' UTR. So this cDNA contains the HA tag but no 3' UTR, and is therefore resistant to miR-159 or any microRNA regulation. By doing this, we found that restoration of TCF7 abolished all anti-proliferative effect of miR-159, confirming that TCF7 is a major target of miR-159. Similarly, restoration of MYC at least partially abolished the effect of miR-159, and this helps us forming this signaling axis from miR-159 to TCF7 to MYC and all the way to the regulation of cancer cell proliferation. In tumors, we confirmed that the expression of TCF7 and MYC are both negatively associated with the level of miR-159. So this is good evidence supporting the regulation of TCF7 and MYC by the plant microRNA. Lastly, we tried to feed not-skid mice with either synthetic miR-159 or plants that are high in miR-159, which I'll explain later. So with the synthetic miR-159, we did R-gluage in a very high concentration because the 25 milligram per kilogram. So this is a typical concentration people use in cancer research when they try to overexpress a tumor suppressive microRNA or to knock down an oncomere in order to suppress tumor growth. And therefore, we used the same concentration. The only difference is in cancer research, people most time inject the mimic or anti-mir, whereas here we did R-gluage. So by doing R-gluage of the synthetic miR-159, we confirmed that we can detect the microRNA quickly after the initial feeding in the GI tract, and if we wait longer, so this is really a very long commitment. We did our daily R-gluage for over a month, and then we start to detect the microRNA in cells and organs beyond the GI tract, including breast and lung and liver, these non-GI tract organs. And we were also able to detect miR-159, although at very low level in the serum, which I will show later. So after I tell you what happened with the tumor. Okay, so we did the same thing this time in mice by first R-gluage with miR-159 mimic, and then we start the tumor cell transplantation into the memory fat pad and continue continuing the feeding of miR-159. We did see that 159 suppressed the size of the tumor and the weight of the tumor, and also regulate the expression of TCF7 and MYC, as well as the proliferation index and apoptotic index in these tumors. In contrast, if we establish the tumors containing microRNA resistant TCF7 by overexpressing this TCF7 cDNA, we lose all the effect of miR-159. And in vivo, again, when we try to understand where the miR-159 came from, we first try the levels of miR-159 in different types of vegetables commonly consumed in the Western countries. So we used the mouse chow as a control, which we hope had very little levels of plant-based microRNA. And apparently we found broccoli as the plant or the vegetable expressing highest level of miR-159. And we were very excited at that moment and thought that we would try to establish a correlation between broccoli intake, dietary intake, and the levels of miR-159, and even the risk of breast cancer in patient populations. However, for many reasons, including the inactive in vivo data I'm going to explain later, well, I'm not pursuing that direction at this time. So we also confirmed that cooking or heating in water of these vegetables do not significantly reduce the microRNA, suggesting the stability. And also, here is what part of the negative data I was talking about. When we choose broccoli as our high miR-159 diet, and cabbage as our low miR-159 or mid-level miR-159 diet. So we use the puree of the vegetable to feed mice for a long time. We couldn't detect significant difference between the two types of food. And we also, the difference between vegetable puree, argillage, and between PBS control is also not significant. Although by argillage of high concentration of synthetic miR-159, we were able to detect some in the serum. So we thought a lot about this, and this is one possibility Andy, my graduate student, brought up. So we think there could be different scenario for the dietary source of a particular plant microRNA. For example, broccoli versus cabbage. So this is totally question marks here. We don't know if this is true at all. We're just using this as an example to explain our hypothesis. In one plant, the microRNA, say miR-159, could exist in high abundance. However, the microRNA may have a specific localization inside of the plant. And for example, in this case, if the abundance is high, but the microRNA is restricted to a highly non-mobile intracellular localization in the food, in the plant, this may also limit its uptake by the GI tract and its further processing and secretion by intestinal cells and its presence in mammalian serum. And in contrast, another plant, if it has low abundance, for example, the cabbage, but maybe this microRNA could be localized to a different compartment in the plant, for example, transport venues, and this may prime the plant microRNA in a position to be taken up by the GI tract cells at a much higher efficiency, and may allow its facilitated secretion of the mammalian cells as well, allowing it to be detected in the serum. However, in order for a plant source to a food source to confer a high circulating plant microRNA in the recipient mammals, we have to have high abundance at the first place and the proper localization of the plant microRNA inside of that vegetable or that plant food. So this is our guess so far. Unfortunately, I don't have the fund and resource to pursue these ideas, but just something I would like to discuss with you. And finally, to summarize what I'm telling you today, we study mostly the endogenous function of cancer-secreted microRNA. So breast cancer secrete these extracellular vesicle-encapsulated microRNA, which can go to different organs, including those organs that would host metastasis. And we believe this would prime the organs to facilitate secondary metastasis. And also, we have realized that there's a very complex crosstalk between cancer-secreted factors and the global environment, including endocrine systems at the whole body levels, and this may, in turn, engage additional organs in the preparation for metastasis. Lastly, as another aspect of the global environment, ingested plant RNA could also play a role in this complex crosstalk mediated by extracellular RNA. Although we only had like one project focusing on this area, I do want to include this in the big pictures because I believe it is happening. With that, I would like to thank people who contributed to the two projects I got a chance to describe today. So Wei Ying is the first author of our MIR-105 paper. Andy contributed and did all the experiments related to MIR-159, the plant microRNA project. I didn't have time to introduce the MIR-122 project, and that was conducted mainly by Miranda. And hopefully I was able to cover a little bit of, both endogenous and exogenous microRNA function today, and I'm happy to take any question now.
Thank you, Emily. We are a little bit over the hour. We have 20 participants, so this is the time for you to make the questions. I think we can allow us to have 5 to 8 minute discussion.
Emily, this is
yes.
David.
Hi.
This is it's very nice work, very thorough, and but I was curious as to the MYC pathway looked like a very important part of the 159 story. How does it fit in? How does it fit into other microRNAs and other transcription factors that feed into that? That one transcription factor, the TCF7, is just one of several involved. Are there other microRNAs, and are they involved in, do you know, in regulating the regulation of cell division in breast cancers?
Sure. Yeah, that's a great question. Absolutely. I think people have reported mammalian microRNAs that can target MYC or TCF7, and unfortunately, I can't remember the exact microRNAs that have been reported at this point. So I think it's a very important point to emphasize that mammalian microRNAs actually, many of them may together regulate the levels of TCF7 and MYC. And based on what I see here, I think, those microRNAs, especially if we're talking about cells adjacent to cancer cells that somehow express altered level of a MYC regulating microRNAs, the effect from the endogenous mammalian microRNA would be much greater than the effect of the plant microRNA we have seen. So something I was thinking about is the stage that plant microRNA may start to take into effect versus when cancer secreted endogenous microRNA may be more predominant in controlling gene expression. So as we know, if these plant microRNA are indeed from dietary source, which our paper couldn't, prove because of the limitation of the mouse model and everything. So if they're from the plant, from a dietary source, then their effect would start, the first day we start to eat plant-based food. 7-month-old, 8-month-old, after you start to have those baby food, and the effect could be lifelong. So I have the feeling that the effect of plant-derived RNA would function more to establish a context or a whole body level, gene expression context or pattern that would either, facilitate tumorigenesis or oncogenic mutations, or for any reason by downregulating certain tumor suppressor gene, or it may in turn suppress the initial upregulation of some oncogenes, for example, MYC. So in this case, although our model didn't really test the tumor initiation stage, my feeling is that at early stage, when MYC is expressed, is overexpressed, or MYC gene is amplified in a very small subset of pre-cancer cells, the effect of miR-159 could be more important at that particular stage during that time window. However, when cancer cells have engaged additional mechanism to enhance the expression of MYC, or I feel the effect of miR-159 would be very, very weak. So this is my 2 cents.
Thank you.
Thank you, Emily. Thank you, everyone. I think it's time to adjourn the meeting. Thank you, Emily.
Thank you so much, Tanya. Thank you, David. Thanks, everyone. Have a nice day.